Hey everyone, my name's Will Patrick. Very excited to be here. This is so cool. This could not have happened. Yeah. I mean, yeah, so cool. This definitely could have happened like 5 years ago. You know what I mean? Like, it's so neat that we're in a place that has a biolab that's about like biohacking, and it's just amazing. My talk it's called personal bio, and that means to me that biology is becoming more accessible. Biology is coming as a part of our lives. That's sort of what I'm going to be talking about, but it's also personal bio because it's sort of like my story with biology. I have a mechanical engineering background, started to do some product design, and I'm now sort of in this world of making hardware products that, sort of exist with biology. So so I'm going to be going through a few projects sort of quickly, hopefully, so we can all go home, but hopefully it should be interesting, provide some eye candy, a little bit different than what we've seen so far. And happy if you want to ask questions during the talk, feel free to do so, or at the end. So biology is becoming personal. So what that means to me is that more people are starting to more types of people are starting to come in. People who are mechanical engineers or designers or artists are starting to think about biology. Biology is becoming so much more accessible, meaning that the tools, genetic and hardware, are becoming cheaper and easier to find, and there's more wet lab spaces that are available. So I sort of see like three trends that are coming in this sort of idea of personal biology, and I've had projects that sort of dabble in all of them in the last 2 years, both in my work when I was a graduate student at MIT, and also recently I was completing an art residency, and I'm going to talk about those three projects. So the first one is from labs to kitchens, meaning when we think about biology, we think biolabs, but as we know from BioCurious and all the different hacker spaces, the space where we do biology is changing, and it is going and becoming our kitchens. The second trend is thinking about using cells, using biological stuff in product design, thinking about the things that we wear, the things that are in our homes. We think, even today with the most startups, we're thinking about stuff that's industrial biotech, things that are in big vats in some factory over there. But something that's also happening is, how are these going to be in our lives? So the third trend is, it's specifically thinking about household products, thinking about what products can we make that would be in the household. So without further, I'm just going to start going into the different projects that are sort of in these categories. So from labs to kitchens. So when I got to MIT, I was new to biology. I walked into a biolab, and I was super surprised to see a lot of really old school tools. And I was working with this thing that was called a syringe pump, which is about $1,000. It has a little LCD display. And it's really just a stepper motor with a linear drive, and it's pushing a syringe very, very slowly. And I'm thinking to myself, like, how is it possible that this thing costs $1,000 and is so terribly designed? And so that is, I think, is so I was very interested in thinking about how can we make these tools more open source and better designed. So the first project was making a syringe pump that was all 3D printable, meaning that you could download the design, bring it, have a MakerBot 3D print it out using sort of all the sort of goody open source stuff, meaning like Arduino processing in order to create a UI to just simply control and program this thing. And so here you can sort of see, you can just sort of put it together really easily. Everything snap fits, screw fits. Operating is super simple. It's very, just sort of what we're more used to than like an LCD display. I was actually really interested in making the syringe pump because I was working a lot with microfluidics. Microfluidics are these devices that allow you to miniaturize chemical and biological reactions. And they're, really interesting tools. But one of the issues with them is typically when you make them, you have to make a master that uses sort of lithography techniques. And so you'd make this master, and then you're actually molding either like silicon or acrylics off of that in order to make your little microchips. You can think about these things as sort of like microchips of biology. Microfluidics, think is like teeny pipes, basically. So 3D printing is this like super promising technology for that, because you could rapid prototype them, make them for dollars instead of hundreds of dollars, make them in an hour instead of having several week lead time. So the rapid prototyping is really exciting. So I was really interested in that. So I explored all these different potential 3D printed microfluidics, some of which are sort of seen there in that little family shot. And here's, so it was like micro mixers. So it was taking two inputs, mixing them in a three-dimensional space. That was made on like a Form Labs device, so a couple dollars. That's a valving mechanism that you could use to route liquids. This one is the smallest scale one that we made, about 200 to 300 microns in diameter, using from Shapeways. Just printed off Shapeways, so which is a website where you can buy 3D printed stuff. So it was again like $5 and several days later, and that's in your wet lab. So it was really cool. We actually just published it, finally got this published about a week ago. What we were actually doing was we were showing that you can use 3D printers to make microfluidics, but we were also running a particular biological experiment, sort of a toy experiment, which was a DNA assembly. If you're familiar with that, a Golden Gate assembly. So taking in like linear pieces of DNA and like one of those little inputs and enzymes in the other, mixing them, having it incubate into the system, and create a sort of an assembled DNA piece, which you would then stick into an E. coli. And just a couple of points about this, like being able to use 3D printing means we were able to rapidly prototype through like all of this designs, explore many design spaces, and that's something with sort of existing fabrication techniques you just couldn't really be able to do. The other thing with sort of 3D printing and digital fabrication allows you to have these CAD design files that are constantly being shared amongst all the researchers. So it means I can be working with 3 different labs. We all have the same common fabrication stuff, 3D printer. We can all be working on it and sort of be sharing and printing out our microfluidic chips. So I think like the conclusion here with like this little trend is that with a lot of these open source technologies, consumer fabrication, you're seeing a lot of what can be made in the wet lab, expensive tools you can actually make using very inexpensive means that can be shared on the web, and this should hopefully lead to more broader accessibility of these hardware tools for wet lab biologists to operate in kitchens, to operate in all sorts of different places. So what I really focused on in my master's research was thinking about how can you bring cells into product design. I got really interested in this like one concept I had, which was what if you could have a pendant that can make Chanel No. 5 sort of on demand. Chanel No. 5 is mostly consists of a bunch of different proteins, like essence of rose, for example, and these might be coming from rose stamens, and they're sort of concocted together, but they're generally all protein based. And so maybe it'd be possible to engineer organisms, people are already doing this, to make those essences or make those smells. And how would it be possible so that instead of just like, again, having the same Chanel No. 5, but have this new experience, it's biologically being produced. So it's just total thought experiment, but you have to think about it. You have to have some kind of pendant that has these cells, that keeps them alive, that is then maybe there's a trigger on your phone that has to do some electronically to introduce like an inducer molecule into those cells, and that like somehow there's like a permeable membrane where like the smell comes out. I don't know. But basically it's this very complicated electromechanical and biological system. And so I was interested in thinking about how these types of systems might be able to be created. My focus on that area was thinking about the containers, like how can we in that pendant, how could you contain the cells? Cells typically are in liquid cultures, so my thought process was similar to those fluidics we were just looking at, perhaps in the pendant or in your other sort of wearable products, you would have these fluidics which would be controlling and containing those cells. So what I ended up making from a master's thesis was this pretty speculative wearable. Speculative meaning what if you could have sort of a wearable that was like a microbiome? So we all know about microbiomes in our gut, do all sorts of different stuff for us. What if you could have one that would make sugar, make food, make all sorts of different things that you wear with you? Obviously, it's sort of in the aesthetic here, if you can sort of see it, is sort of inspired by the intestines. So it's obviously speculative, but like we were just trying to think like what would that even start looking like? So for the design process, like when we start thinking about these fluidics and we have them conforming to the human body, it starts to be hard to do that in a normal CAD platform. So we worked on doing like computational approaches in order to grow these types of fluidic networks, meaning like all the channels that would be on the wearable, conforming to the human body, 3D printing them using multi-material 3D printing, and lots of interesting, we had to do lots of interesting things about how do you actually 3D print these negative channel spaces, but that's for another time. And what we were actually designing it for was different cyanobacteria strain that it was engineered by a collaborator lab to greatly produce sucrose. So sort of that idea of this sort of wearable that is creating sugar, creating food for you. And so we actually created these transparent regions. I don't know if you could really tell, but on the piece there's these transparent regions where we'd have the cyanobacteria that were growing the sugar. And so in those areas, the light could come in and be used for photosynthesis. So we, this is sort of what it looked like when we actually put it on somebody. It's not, nothing says wearable like wearing it, I guess. And there's a whole video documenting this project. I'm happy to share it with you guys if you're interested. But I'm just super fascinated. This is not like what's going to happen next year. This is not what's going to happen in 5 years. But starting to think about, we can engineer organisms. How might they augment our human biology itself and through the products that we're wearing? I think is a super fascinating direction. And finally, I want to talk about sort of household products. So I did this residency, as I mentioned, at Autodesk just this past fall. And I was trying to figure out what to build. It's this really amazing workshop. It's on the pier. If you've never been there, you should totally go. They have all the most amazing fabrication tools in the world, literally. And so you can sort of do whatever you want. At the same time, when the program started, there's this amazing paper that came out, probably many of you have seen, from the Smolke group in Stanford, where they showed opioid synthesis in yeast, meaning going from glucose to hydrocodone. And so this is to create an alternative supply chain to poppies for making opioids. And so I was like, this is really interesting. I'm very curious about, if you could have yeast that produce opioids, why not be able to do this at home? Why not be able to have distributed sort of pharmaceutical production, sort of getting these themes that we keep hitting back to, it seems like, today. So I was really interested, and as sort of coming at it from definitely a design perspective, what would that look like? What would this system that is at home that's making drugs for you, how would it work? What would it look like? And so that was where I explored. This is what I ended up making. I called it Pharma. And and so how did it work? So it started, the organism of choice that I used was spirulina. Spirulina, as you know, is like a blue-green algae. It's commonly eaten as a nutrient. And the idea was maybe you could engineer spirulina, maybe similar to yeast, in order to produce opioids, to produce cannabinoids, to produce omega-3 fatty acids, maybe lots of different things, maybe personalized. But they're produced in spirulina, and then you can just consume the spirulina. Because one of the sticking points I kept getting to in my head is like, how are we going to do this distributed pharmaceutical production? You make it in yeast, then you have to purify it somehow, then you have to eventually somehow take that. Well, if it's being produced in spirulina, then maybe you can just directly consume it because it's a consumable. So you have spirulina. The idea was in this hypothetical product, you could have Keurig cups that come in with your sort of drug of choice, your starter culture, if you will, if in thinking about kombucha, and you drop that into your bioreactor. The bioreactor I designed was a sort of airlift photobioreactor. It had this centralized light column. There's an aerator. There's all these pumps on the inside in order to pump stuff out. The reason I was pumping out is I needed to filter it. So spirulina is a very large organism, and so you can actually just filter it. So I used a filter in order to filter out the spirulina, and then a pump to recirculate it. Also, as a part of the sort of documentation I made, so how do you get the spirulina, how do you actually eat it? So this was a gel cap filler, so a pill maker in order to create the drug that you're going to consume. So that was the end product. So how did I get there? So I had all these different design iterations that I went through. For me, it was really important to consider sort of the aesthetics. What does this really feel like? Again, coming at this from more of a product design angle. So I immediately thought of sort of classical, iconic sort of kitchen appliance designs, looking at Braun, coffee makers, et cetera, things like that, in order to start thinking about the cylindrical approach. But also the cylinder really came out of the requirements of a sort of photobioreactor. And just other technical bits in case you're curious. Yeah, it's an internally illuminated airlift design. It's got all this crazy stuff on the inside that barely fit in our like 3 different pumps, Arduino, et cetera. And I was able to program it from the outside. And again, in case you're curious, being that I was at the Pier, I wanted to use really great tools and really great materials, stuff that you would actually find in a real consumer product. So it was all like CNC milled aluminum and CNC milled Corian. Corian's a common material on a countertop. This project culminated with a show that we did in November, and it was really cool for me because I'd never really done an art show before, but it was also really exciting because I was going to be able to sort of see what people's reactions were. I sort of came into the project feeling very torn between like, I'm super excited that like people are going to be able to like take control of pharmaceuticals and be able to like make their own stuff, and also like very terrified at like the possibilities of like Breaking Bad and like people becoming addicted to opioids. And so I like personally had that tension, right? And so I wanted to create a project that sort of shared that internal conflict I was having. And so actually, I don't know if you can tell, but the aesthetic I was going for is sort of trapped between something that seems like very normal and understandable and something that seems sort of cold and futuristic. But anyway, so getting to the actual show, I was really interested to see what people had to say. And so it was really fun just like to talk about it and get people's opinions. And there was also an Instructable that I put on the web where, in order to get sort of public online feedback. And there's all these different types of people and their like feedback on it. So there's some that were like, this is really cool. I like to brew stuff at home. I like make kombucha. Like, this is awesome. There's another group of people who are like sort of like the Soylent crowd who are like, I just want to like engineer everything in my body. And like, this is really neat. I'm like, I want to personalize everything. Like, I'm a total engineer. And that was really interesting. Other people other people love spirulina, which I didn't know about. Like, other people like love spirulina, and are like, I don't care about pharmaceuticals. I just want spirulina. I was like, oh wow, maybe I should do that. And then other people were like, this is totally scary. Other people were like, can you engineer spirulina? And then other people were like, I just don't get it. I want to buy it. So anyway, I think it was really. I don't want to make it myself. I'll just buy it from the company. Like, I don't really care what you're talking about. You're stupid. So, so it was really interesting for me, and that was the point of the project, was to like provoke all of that, was to provoke all of those viewpoints. And like my thought process on this particular project was, there's a huge disconnect, I think, generally about what's going on in sort of the research community in terms of the development of genetic techniques and the possible implications with the public. And so I think there's roles for these types of projects to play with just provoking and creating all of that discussion. And so that's what I was really trying to go for in this project. I think that's it, everyone. Really appreciate your time. Thanks for having me, and would love to take any questions.